Well, it's a pleasure being here and having the opportunity to speak about an area that is of great interest to me. Um, how do I come to the laboratory and then say that I don't actually have patients? I'm a laboratorian. I've worked in national reference labs, and before that, I was an academic by background. I am a rheumatologist who studied viral pathogenesis of rheumatic diseases, and very early in my career had perhaps back in 1988 when the CLIA regulations came out, had a CLIA laboratory directorship studying viral etiologies of arthritis. Clearly, we have neurologists and rheumatologists here. I spent 13 years at Quest Diagnostics as medical director for immunology R&D, developing the diagnostic tests in the autoimmune neurology portfolio. And I've also worked before coming to my current location, LabCorp. I had worked at Euroimmune, which is perhaps the largest manufacturer globally of automation solutions, kits, reagents for autoimmunity and infectious disease. So have an opportunity to see the development of laboratory testing firsthand, and as an immunologist, I'm very comfortable looking at immunological methods in diagnosis of organ-specific diseases. And perhaps my most recent contribution, which I would recommend to you forthcoming, is the Manual of Molecular and Clinical Diagnostic Immunology, which I edited the organ-specific autoimmunity section, and there's a very nice chapter on neuroimmunology that Dr. Grouse wrote for that section. It should be out in October, hoping. So what are we going to do today? We're going to discuss the role of the laboratory in aiding the diagnosis of autoimmune neurological diseases. We're going to describe the strategy for testing autoantibodies in autoimmune neurological diseases and explain the methods to detect autoantibodies in autoimmune neurological diseases. And so we're going to focus on antibody mediated or assisted um, uh, diseases in this instance, simply because as a member and chair of the board of directors of the Autoimmune Encephalitis Alliance, that is our focus. And so I wanted to focus our talk today on those areas. So we like to think of the autoantibodies associated with these diseases that we'll discuss today as being targeting either intracellular antigens or cell surface antigens. And I think, as many of you already know, those antibodies that are associated with targeting intracellular antigens are considered biomarkers of disease rather than being specifically pathogenic. What we do suspect is that they help direct cytotoxic T cells to target the cells that contain these antigens, and which may, through the normal processing of proteins, may ultimately at times be presented on the cell surface in the context of MHC as peptides. And so the testing tends to focus on them as peptides. In contrast, the cell surface antigens are typically antigens that are essentially cell surface receptors involved in synaptic function. Um, and so in that sense, the antibodies are pathogenic because by modulating cross-linking receptors and modulating off the surface, they reduce their density on the cell surface. And so there are a number of these that you're well aware of that play critical roles in synaptic function. So the other difference that these tend to show, and we're not going to really speak about too many clinical aspects or therapeutics, but certainly the intracellular antigens as biomarkers tend to be resistant to therapy, while suppression of antibodies targeting the cell surface synaptic receptors tend to be therapeutic, in that if you remove the antibody, the receptors have a chance to be regenerated on the cell surface, and therefore return function. So why would we make antibodies to what are essentially self antigens? Well, one is molecular mimicry, where the immune response to an infectious agent or an environmental factor cross-reacts with neuronal tissue. And perhaps we see an example of this in this space, where individuals with herpes encephalitis were treated with antivirals, improve, and then a couple of weeks later return with what appears to be a return of encephalitis, and it turns out that it's NMDAR1 antibody. So that's an example where we may see molecular mimicry through exposure to an infectious agent. There may be other causes for loss of tolerance, such as an anergized or quiescent B cell clone that produces a pathologic antibody, or awakens, or that a pathologic clone arises through somatic mutation. Also, and for those of us here who are interested in oncology, when cancer cells progress, they often express developmentally inappropriate antigens or novel tumor antigens, and this may be seen then as novel to the immune system and mount an autoantibody response. This is a process that we like to see happen as part of our tumor surveillance. And so there are instances where one may have a presence of an autoantibody to neural tissue that's often associated with an underlying malignancy, such as a small cell lung carcinoma, but yet a tumor is not found. And there are cases where those individuals, when they come to autopsy and have a very careful dissection of a lung, you may find a small nidus of tumor. And so that may suggest that tumor surveillance has suppressed that tumor's progression. So we also have non-neuronal tumor cells that express neuronal proteins. And so normally those neuronal proteins are hidden from the immune system, but again, they're expressed, and as part of tumor surveillance, the antibody response may arise. And again, in many cases, no tumor is found, suggesting a resolution of tumor idiopathic autoimmune mechanisms. And so there's been an attempt in the literature to categorize these autoantibodies as those antibodies that are highly likely to be associated with tumor as part of a paraneoplastic syndrome, or those that may be moderate, or those that have low risk of being associated with tumor. And so when we think about what the antibodies actually target, they target neural cells, and I'm using neural broadly, so meaning neurons as well as glial cells. And they may target any of a number of different structures within that cell: nucleus, cytoplasm, cell surface receptors, axonal covering. And that becomes important in beginning to understand the patterns of staining that we see in indirect immunofluorescence or neural tissues. And ultimately, the mechanism of injury could be disruption of intracellular function, cell surface molecular interactions, neurotransmitter communications, or we can see cell depletion through antibody-dependent complement fixation or antibody-dependent cytotoxicity. So how do we approach testing? So the consensus is that one needs to do a two-tier test. So one either first looks at indirect immunofluorescence assay and looks at the pattern of staining on neural tissue, what structures in the neural tissues are stained, what cells are stained, and within the cells, what parts of the cells might stain. And then determine based on the pattern will give you some guidance as to what specific autoantibodies to look at, would be then to determine the reactivity of specific target antigens with specific target antigens using any number of methods that have been used. Membrane-based methods, so immunoblots or a updated version of immunoblots, Western blots would be a line blot, or looking at transfected HEC293 cells that express the antigen. CHO cells have also been used. And then radioimmunoassays, typically using brain cell membrane fragments that contain a receptor and labeling those particular receptors with a neurotoxin that specifically binds to that receptor, and the neurotoxin being radio-labeled, and then doing immunoprecipitation. More recently, as the advent of analyte-specific tests have become more broadly used, and there are laboratories that may want to first look at the reactivity on specific antibody tests. But it's important to confirm what you find by looking at the pattern of immunofluorescence staining on neural tissue. And I say immunofluorescence, that's widely used. There are some laboratories that are using immunohistochemistry as an alternative form of labeling. So the challenge is to be broad, and I would bring your attention to this wonderful monograph by Darnell and Posner on paraneoplastic syndromes, which is very extensively documented with lots of cases and examples, and also a lot of information about what immunofluorescence should look like, what the individual antibody, what structures they stain, what antigens they target. So I would recommend this reference to you. And what they said is that ideally, immunostaining should be done against several CNS tissues. And in addition, Western blots or ELISAs, the prominent analyte-specific assay methods that were available back in 2011, should be performed using both against clone fusion proteins as well as extracts of neurons. So they take a rather broad approach to testing. And so the traditional method that began, that really was helpful in identifying some of the first autoantibodies to neural tissue was an indirect immunofluorescence. And I think just about everyone on the call will be familiar with the basic principles of the assay. So basically, one puts a tissue, or ultimately cells, on a slide, and the substrates, the tissue substrates have been both rat and non-human primates. More recently, come back with the patient's serum or CSF, and if there's antibody there, it'll glom on to the tissue. You wash off which is not specifically bound, and then you come back typically with a GOAT anti-human IgG that is conjugated to a fluorescein. So this second antibody is often called a conjugate, and you might hear me lapse into that term. And it's fluorescinated with a fluorescein marker. And then you look under a fluorescent microscope for the green staining. Now, early on, laboratories used rodent tissue substrate. And so one would take rodent tissue and would stain and look for pattern of staining both for the structure, for example, an MDR, which stains the molecular layer, the neuropil, and we'll look at some of the structures in a little bit, and other staining patterns. And so you see a number of different staining patterns that are associated with specific antibodies. And so in using rodent tissue, which has been the early mainstay of IFA in this area, one has to ask, does the rodent tissue target an antigen with sufficient homology to a human counterpart to be recognized by a patient antibody? Early on, one of the concerns was the presence of antibodies to glycoproteins and subsequently antibodies to non-specific anti-nuclear antibodies, because some of the first autoantibodies seen in paraneoplastic syndromes were neuronal specific nuclear antibodies. And so how could you distinguish those from the non-specific autoantibodies that stain everything? And so the early attempts, and some laboratories still do, will do a serum pre-absorption. And so you have to ask the laboratory when they're screening serum that has been pre-absorbed, at what concentration are they doing the screening, because there's a sufficient absorbing protein in the serum to make undiluted serum blurry on an IFA. So serum has to be diluted out a bit further before you can screen. And then ultimately, what is the screening titer? Because you want to know when someone is called seronegative, that you know at what titer they were considered seronegative. Now, more recently, and I'll talk about this technology simply because it's one that's been adopted by any number of national reference laboratories. This is a biochip technology in which a tissue is sectioned and placed on a glass cover slip, and it's fixed to the glass cover slip with an adhesive that partially fixes the tissue. And I don't know what that is because it's the manufacturer's secret sauce, but it fixes the tissue partially, and it's a gentle fixation. It sticks on the glass cover slip, and a technologist looks under the microscope at this material, putting over the tissue a virtual grid, and looks at each sector to see if it meets quality control parameters for the quality of that tissue. And then a laser is used to cut the chips and to take the ones that have been approved and put them into a microscope well. And you can do a similar thing with cells that have been fixed on the cover slip. And cells may have been transfected to express a protein of interest, a cell membrane typically, a cell membrane receptor typically, and then the same process is done. So this gives you standardization in terms of the quality of the product and extreme miniaturization. And so this is what it looks like under the microscope as these are examined and or sectors X'd out and not used in the manufacture of slides. And so you can put multiple chips in a slide, and here's a typical mosaic of primate cerebellum, rat hippocampus, and sural nerve. And then a number of other tissues are added so that you can begin to understand the differential staining pattern of some of the antibodies. So for example, Hu and Re are very similar on the monkey cerebellum in terms of staining the granular layer in the cerebellum and the neurons. But the difference is that Hu will stain the myenteric plexus in the small intestine, Re will not. In the kidney, for example, we have that in order to detect the very rare antibody ANA3, which will stain podocytes in the kidney. The testis helps you identify the difference between MA1 and MA2 because MA1 will stain the sperm precursor cells in the nucleolus, whereas MA2 will not. And they'll both stain the nucleolus and cerebellum, for example. And then pancreas to differentiate GAD65 from amphiphysin, which look very similar on the neural tissues, but GAD65 will stain the pancreatic islet cells and amphiphysin will not. And many of the mosaics will include a HEP2 cell, which is the traditional substrate for doing anti-nuclear antibody testing to look for non-specific staining of cells. And this is not just nuclear staining, but also cytoplasmic staining as well. And one can see then if the staining pattern is the same or different. So even in the presence of a nonspecific ANA, it's possible typically to differentiate a unique staining pattern that's neuronal specific or neural specific. So the methods that are used is the same as you would use for a tissue. For your mosaic, you would add patient serum or CSF, wash, and then come back with the GOAT anti-human IgG conjugated to fluorescein, and then look. So here's an example of some of the structures that are looked for in the laboratory. This is cerebellum, and typically you have lamina alba, which are nerve axons. And, occasionally, for example, you'll see a myelin antibody when in high titer light up the lamina alba. The next is the granular layer, which has any number of neuronal cells. And so you can see this dense packing of neurons. There's around the outer portion, there's a series of Purkinje cells. These are, as you can see here, very large neurons, a pyramidal cell in shape with a single apical dendrite that, as it moves into what's called the molecular layer, arborizes and interacts. And so these are structures that one looks at in the cerebellum, the structures, and also what parts of the cells are staining. Here's an example of the hippocampus, and you can see we can look at hylar staining, their molecular layer, and what's often called the neuropil for staining. And so, these are typical areas of staining that one looks at for those autoantibodies involved in limbic encephalitis, for example, like NMDAR2. So these are some of the patterns that one can differentiate by looking. And the secret is in the details. And so, for example, you can see Purkinje cell staining, which looks like a string of pearls. And, Yo or a PCA1, typically the cytoplasm will be stained and the nucleus will be spared more or less. And the PCATR or dinner, there's a tendency to stain like this, but also the base of the apical dendrite, and ITPR and CARP8, for example, will go on to stain the dendrite and the arborized nerve axons. And so those and ARG or Rho GPase 26 antibody, for example, will stain and gives you what looks like a medusa hair, a medusa head. And so this group, some of these antibodies in this group are called medusa head antibodies. And so those are very telltale patterns. Neuronal nuclear antibody typically will see staining in the granular layer. We'll see some staining in Purkinje cells in the cytoplasm. And so you can follow those as well, clearly different. Neuronal nucleolar staining, you can see, for example, in MOT1 and MOT2 Ta, and then intracellular synaptic staining like GAD65 and amphiphysin, and then intracellular CV2, which stains in other areas. So these different patterns looked at in detail gives you a clue as to what antibody you may be dealing with. And so here's a couple of examples. Here's an example of Hu antibody, and you can see the granular nuclear staining in the cerebellum. You can see some similar staining in the hippocampus, really not much, nothing in the nerve. And then if you look in the intestine, and we'll look a little closer, you can see the myoteric plexus. So let's take a closer look. So in the intestine, just to review some brief anatomy, there are two muscle layers. There's a circular muscle layer that is inner, and then an outer longitudinal layer, and between them is a plexus of nerves, a myenteric plexus. These are non-medullated nerves. And if you look at trying to differentiate Hue and Re, ANA 1 and ANA 2 on cerebellum alone, there's not, they're very similar and you really can't differentiate them. But here is an example of the monkey intestine. Hue will stain the myenteric plexus and Re will not. So that is an aid in further differentiating Hue and Re. Purkinje cells, again, we talked about that a little bit. Here's an example of a string of pearls. And in the sural nerve, you can see the staining of the nerve sheaths with myelin. MOG staining looks a little bit different, a little more circular and granular. So you can differentiate, for example, myelin and MOG, at least be suspecting what the antibody is. Here's another example of tissue differentiation. So again, on rat hippocampus, GAD65 and amphiphysin look very much the same, but in the pancreas, you can see that GAD65 will stain beta cell, pancreatic islet cells, and amphiphysin will not. So in many of the laboratories that are using these this approach of mosaic testing, there are fully automated imaging platforms to help to capture those images. And what's important, I think, for clinicians to recognize is that laboratories will archive these images. So if you have a question about your patient, about what was seen on the IFA, if you want to see what the images are and learn about those patterns, reach out to the laboratory and ask to share and to review those with the laboratory director responsible for the testing. I think that's a very helpful educational tool, and it may provide you with some insights in terms of your patient. So the indirect immunofluorescent patterns are suggestive or definitive for a number of antibodies, and so it's important to have the laboratory let you know what they're seeing. So once you have an IFA pattern suggested, it's important to confirm it by a monospecific antibody test. And so there are a number of single analyte tests that also use IFA, cell-based assays, in which a cell has been transfected to express the protein of interest. And so there are fixed cell versions in which the manufacturer, again, fixes the cells to a slide, or live cell assays in which the cells are transfected and then essentially put through a flow cytometer to identify antibody binding. There are membrane-based tests. The Western blots have fallen out of use because it's difficult to know that if you're looking at for a particular band at a particular molecular weight, that is the protein. There are other proteins that share similar molecular weights. And so these have been replaced with line blots, and we'll talk about those. And then finally, particularly for the neuromuscular junction antibodies, radioimmunoassays. So, the transfection methodology, I think most of you know, one identifies the DNA of interest, you synthesize sufficient amounts, put it into a plasmid, and then express the plasmid in your cell of interest. Very typical in the industry is the manufacture of HEC293 cells that have been transfected. They're fixed on a slide, and then you do an indirect immunofluorescence as you would for a tissue. So there have been a number of these and that have been done in a mosaic format. You can put a single chip in a well and have a single assay. And so the approach of doing this as a mosaic has some advantages. And so the advantage is that you have internal controls. So here's an example of a number of autoantibodies that will target the hippocampus. And you can see that in the hippocampus, the patterns are very similar. The cerebellum also very similar. And so you then need to do a single analyte assay. And so here's an example of someone with NMDAR1 encephalitis. And so the biochip with NMDAR1 expressing HEC293 cells shows staining, particularly in the axons and the base of the cell. So that's positive. And the others on the chip are negative. And the same thing happens with the others analyte-specific assays. So these provide internal controls for testing. The other method that has been popularized recently is a Western line blot. It's a Western method, but the antigen that's purified from tissue or is a recombinant antigen and purified is then placed on a membrane that is optimized for binding of that particular protein. And you can also put a standard amount of antigen. If you use a Western blot and you're taking cell lysates, you really don't know how much of the antigen of interest is on the slide. But here you can standardize. And you can also end up doing semi-quantitation by looking at the densitometry of the band that is formed. And that band is formed in the following way. You put the anti-patient serum or CSF on the blot, you let it incubate, you wash off what doesn't stick, and you come back with an antibody conjugate, but instead of fluorescein, this is alkaline phosphatase. And in the presence of color developer, it will develop a brownish band at the site. And so this is an example. What one does is take these, and the labeling is a little off on the screen, but one then takes the chip and puts it on a long plastic backing strip so that one can take the whole strip, put it into a slot well, and do the steps for the antibody addition. And the buffer is a universal buffer that has been optimized to work with all of those on the strip. And so again, this is also automated, so you can avoid human error in terms of additions, adding amounts at each step, and timing. So the radioimmunoprecipitation assays essentially take a neurotoxin that is labeled with I-125, and the neurotoxin is selected to be specific for that channel protein. And that channel protein it takes up the neurotoxin, and then one adds patient serum or CSF. Typically it's serum because we're really not looking for neuromuscular junction antibodies in CSF, and one then does an immunoprecipitation, washes, and then measures the radioactivity and the immunoprecipitant. The voltage-gated potassium channel, and these the sources for these channels are typically rabbit brain lysate plasma membrane. And so you're bringing down not just the channel, but everything in the membrane fragment. And for the voltage-gated potassium channel, the neurotoxin is alpha-dendrotoxin. comes from Mamba steaks and has been used extensively. But the VGKC assay has fallen out of favor, and you'll find fewer laboratories doing it. One, because if you have an alternative, you don't like working with radioactivity. But more importantly is because what the target of the antibody was LGI1 or Caspr2 associated with the VGKC channel. And so now there are direct assays for LGI1 and Caspr2 as cell-based assays that have replaced it. But there's still radioimmunoprecipitation assays, or RIPA, being used for voltage-gated calcium channel, both PQ type and N type, and you can see that these are differentiated by the different conotoxin that is being used. These are toxins that come from marine cone snail, and so there are different versions that are specific for these two different receptor types. And that's how those receptor types were identified and discovered, because of differential binding for neurotoxins. Ganglionic acetylcholine receptor alpha 3, that's an antibody that's seen in autoimmune dysfunction. It uses an Ecuadorian frog skin toxin, epibetadine, and then the acetylcholine receptor uses alpha bungarotoxin. And there are 3 different tests that are used for acetylcholine receptor. One is a binding assay, in which you simply look at antibody that will bind to the receptor after you've added the bungarotoxin. And there is another assay that looks at the ability of antibodies to block the ability of the bungarotoxin to bind to its receptor site on the channel, and that's a blocking antibody. And why would you do that? Well, it's about positive in about 50% of individuals who have a binding antibody. So if you want to reassure yourself that your positive binding antibody is a real result, you can do a blocking assay. And there's a modulating assay in which cells that have acetylcholine receptor on their cell surface are incubated overnight with antibody. And if there is modulating antibody, namely antibody that will cross-link the receptor and internalize the receptors such that there's fewer receptors on the cell surface, the next day you come back and you look to see how much radioiodinated bungarotoxin will bind. That gives you a measure of modulating antibody. And so if you suspect someone may have a myasthenia gravis, and you have a negative acetylcholine receptor binding assay, then you can consider it would be a modulating antibody is present that that would interfere with the ability, not necessarily to bind as you would directly, but rather modulating the receptor off the cell surface. So those are 3 of the acetylcholine receptor assays. So I think the take-home lesson here is, 1, you need to know your lab. You need to know what your lab is doing, what antibodies are being tested, what methods are used, and what's always tested, what's reflexed, and when. And the important thing is get to know your lab director. In the old days, we would just walk down the hall and talk to the pathologist running the test. Now, these tests, which are somewhat esoteric, are typically sent out to national testing labs. And so you use the phone. That's your walk down to the hall or to the basement to talk. And I think in the laboratories, you'll find that your laboratory directors are very willing to reach out and discuss patients and to be a resource. Remember that, for IFA images, for example, they're archived. So if you have an interesting case, you want to do a case report or a paper, you can pull these out and illustrate your findings. The other thing to be aware of is that clinically relative positive antibodies may be found that may not be ordered. And laboratories are often constrained in that they can only test what you order and they can only report what you order. When I found this circumstance in my former life as a medical director for immunology, responsible for this testing, I'd pick up the phone and call the clinician and say, "You have you have something that you need to be aware of," because I think most physicians would agree with me that there's an ethical responsibility to let the provider know that there may be something else going on. And so that's something you have to be comfortable with, and why I would encourage you to reach out to medical directors, whatever the lab is. And I'm sure, because I know them, they're willing to be helpful. So the last question is, one of the last questions is serum or CSF? Certainly for the intracellular antigens, antibodies in serum is typically adequate. Aquaporin 4 is the cell surface antigen, but again, serum is often adequate. For the cell surface receptors associated with limbic encephalitis or hippocampal staining, CSF and serum is optimal, much as Darnell and Posner would argue, because as an example, in MDAR1, 14% of patients roughly who have an MDAR1 encephalitis have no antibodies in the serum, but only in the CSF. So you need to think about the case and think broadly about CSF and serum. And one of the other advantages of CSF is that, unlike serum, CSF will not have common autoantibodies that are not specific to neural tissue, such as thyroperoxidase antibodies or non-specific ANAs. Now, patients don't always follow the rules. That's one of the challenges of being a clinician. And the laboratory is extremely helpful. I'm a laboratorian. I think I will tell you that. But ultimately, the diagnosis rests on the clinician and an integration of all the data, the clinical data, the history, the exam, the neurological exam, of course, most important, imaging, and all the laboratory tests. So it becomes important to think broadly. But when I say that patients don't always follow the rules, I'm struck by the data that was presented by Winfried Stucker back in 2015 at the Dresden Antibody Conference. Winfried Stucker was the founder of the Euroimmune Clinical Immunology Laboratory in Lübeck. And this served as a reference laboratory in neuroimmunology for all of Europe. And they looked at over 16,000 samples that had been sent to them. And their clinical lab tested the tests that were ordered. But what they did for the study was to test the samples for everything they had, both on the clinical menu as well as in the R&D pipeline. And when they did this, they found a single positive antibody in almost 14%. 0.5% had two antibodies. And so what I found most interesting was that when they had a positive, it was positive for what the doctor ordered in only 53% of the time. And another almost 47% of the time, it was positive for something that was not requested. And what I looked at the time back in 2015 is what was commercially available, what was available and currently being tested in national reference laboratories. There were 22% of the positives were positive for something that was not yet available for routine testing. And another 13% was positive on tissue, but there was no specific analyte confirmation. So these were what were called UFOs, or unidentified fluorescent objects. And that UFO population has been a wealth of opportunity for discovery. And so if one were then to look at, for example, a typical GAD antibody pattern, and it was confirmed to be GAD on a monospecific test, or a GABA A B receptor antibody, and it was found to be positive, that's fine. But what if you had a GABA B antibody type pattern, but it was not confirmed by an analyte specific test, or you had a novel pattern and no known antibodies were identified. So what one then does is attempts to identify what that is as an antibody discovery activity. And there have been two approaches that have, well, actually three that have been involved. And so the first one that had been used to discover many of the antibodies that we're now aware of is immunoprecipitation of either brain lysates or um, um, laser dissected sections of cryosections. And the tissues are then homogenized, and the uh, antibodies are added from, presumably from the serum or the CSF from a patient in question who presented that UFO. And then a secondary antibody is added to pull down the to pull down the patient's IgG, and typically it's on a magnetic bead so that you can wash it, pull it out, and then elute that and put it onto a gel and identify the bands that represent proteins and send them off to MALDI-TOF and identify what those proteins may be. An alternative approach is to use a phage library display in which a library of DNA that can be extracted from a tissue such as brain, or it can be a synthetic um, library with random sequences that are placed genetically into a bacteriophage. And here's your typical bacteriophage, which has, like a lunar landing, 5 pili or legs. And you can put the sequence of interest into that, into the pili. And so if you put patient's serum or CSF on a plate with allowing the antibody to adhere, it'll capture that bacteriophage, and then you wash off what doesn't stick, and then you come back and amplify, grow up more of that particular clone, and then ultimately use DNA sequencing to sequence what the peptide is and then identify it that way. And a third way, which I don't display here, but a third way is to use a proteomics display of protein arrays and look for antibody that'll stick. So there have been a host of UFO target identification. Many of the antibodies that we now see have come into the commercial space that are available in reference lab have been identified in very similar ways. And so the list of antibodies keeps growing. And the big challenge is to identify an antibody in perhaps a single case and then ask, is this something that's unique to the patient? Is this clinically relevant? Are there other cases? Is there a phenotype that we can associate with this antibody? And that's the challenge and where we are. And so just in terms of background reading, I would again recommend to you Darnell and Posner. These are two references, 2 and 3 are references that talk about methodology. And then these last 2 references talk about methods for identifying novel autoantibodies. And I'll rest there and stop sharing and be happy to take any questions.